Great. Thank you, Keith. Before I get started, I want to thank Keith for inviting me and Terry for taking care of all of the magical wizardry that goes on behind the scenes. So today I'm going to talk a little bit about this idea of putting genomics to work. And for over 10 years now, we've been saying that host genetics plays a role in determining mountain pine beetle resilience, but we haven't really had all of the information to prove to everyone that it does play a role. And a couple of weeks ago, Dr. Rhiannon Peary gave a lovely talk on why genes matter for this mountain pine beetle system. And so I'm not going to go over all of the things she said, but if you were not able to be at her talk, there's a couple of key things that I'm going to pull out that demonstrate that genes matter. So the first off is this image here. So what this is a summary of a number of genetic markers that are different between co-evolved hosts and naive hosts. And so here we see that in the area where mountain pine beetle has historically been, the allele frequencies are quite different at these markers than outside of this range. So we can differentiate trees that have co-evolved with mountain pine beetle versus trees that have not, using genetic markers. So this is our one little bit that says there's coevolution occurring and it does matter to the genome of these trees. The second piece that I wanted to pull from Rhiannon's talk is this really cool opportunity they had. So they have these stands where it's an epidemic has gone through, lots of trees have died, some trees lived. So if we take their offspring and we compare it to the offspring of trees that died, there are genetic differences. And so I've highlighted in these two boxes those main differences is that we've lost rare alleles. So the x-axis is the allele frequencies and the y-axis is just the number of markers. Alleles that are really rare are being lost when we have an epidemic. So that says that the gene pool of the trees exposed to the epidemic is different than post-epidemic. So trees are going to be different after an epidemic happens. And then the third thing is that we can identify genetic variation that's associated with the tax status. And this is independent of clinal or environmental associations. So Rhiannon a couple weeks showed that 26 markers are associated with the tax status. So trees that were attacked have a different allele than trees that weren't attacked. And this is significant. And so all of these bits of information tell us that genes matter. And so when we talk about host genetics and how it plays a role in determining mountain pine beetle resilience, we should really be thinking about how we can incorporate host genotype in our models. So if we're going to model a mountain pine beetle outbreak or spread dynamics, the usual suspects are climate, the distribution of the host, which will include both the density of host as well as the age structure of the host, the reproduction of the beetle the year before, and then we're arguing we need to include host genotype. And this brings me to a really important question. How do we operationalize this information? So we've shown you that genes matter. How do we use this information going forward and apply it to mountain pine beetle management? And so obviously the first thing that likely comes to many of your minds is tree breeding. And tree breeding is an excellent option for this type of genetic data we're finding. We can breed trees with more resilient alleles in an individual and create more resilient individuals. Tree breeding does take time. So this is something we need to think about as we go forward. Also, I'm not a tree breeder, so I'm not really going to focus on tree breeding. The other thing we can do is assisted migration. Rather than taking all of these years to grow new trees and develop a more resilient tree, let's take seed that already exists and plant it in areas that are going to be more at risk of mountain pine beetle. It's kind of, assisted migration It's been talked about lots in the forestry management realms because of climate change. So maybe we could use it also in a mountain pine beetle context. So both tree breeding and assisted migration are great approaches that focus on replanting strategies to increase future resilience. So both great options to put what we've learned to work. But what I'm going to focus on today is the question about what about our existing forests? How do we use the information we've gained over the past 10 years about the pine and their genomics to manage what's on the landscape? And here we go, genes in space. So this is what I'm going to focus on. And this is really where I get excited about, because my training, while I'm, a population geneticist, my base training was in landscape genetics. So looking how genes vary across the landscape and why they vary across the landscape. And can we predict how they vary across the landscape? So that's what I'm going to focus on in this talk. And I'm going to use two main examples that demonstrate how we can operationalize this genomic information. So the first one is an example you've seen before. So we're translating genetics to the landscape, and we're going to use this example of the lodgepole jack pine hybrid zone. So this hybrid zone, we know it extends from Alberta up into the Northwest Territories, but based on these kind of rudimentary, really useful, but rudimentary maps, we don't really know the spatial extent of the hybrid zone. And so over the years, we've been using genetic data to refine this. And the most recent refinement of this was done by a master's student, Ian Burns. And what Ian did is he looked at genetic ancestry. So we used genetic markers to estimate ancestry, where an individual could be completely lodgepole pine, it could be completely jack pine, or it could be somewhere in the middle of that. It could be a 50-50 hybrid, so an F1 or an F2 hybrid, or it could be some sort of back cross. And the relationship of that with geographic and climate variables developed a really strong prediction for ancestry. So if we look at elevation, latitude, longitude, climate moisture, and summer heat moisture, we can predict what species will be on the landscape and what level of ancestry it has. And so I've just shown you here the kind of model metrics showing the area under the curve, and this is a very excellent model with a really high predictive success. And so what we've done is we've created an entire prediction across the country, and this has been used in an operational setting. So what this is we have pure lodgepole pine is predicted where it's dark green, pure jack pine is predicted where it's light green, and hybrids are predicted where it is orange. So you can see that this hybrid zone is quite variable and quite complex. It's not, a nice simple straight line. Some areas it's wider than other areas. So it's not spatially uniform. And so this has been used in an operational setting. So it has been used by Barry Cook and Alan Carroll when they were looking at predicting the risk of mountain pine beetle and looking at eastern spread in the forests. I supplied this data to them because it could be an important variable in their model, so they can test whether or not that really makes a difference in the spread of mountain pine beetle. We can also use it for other things. We can actually use it to predict how the distribution of these species might change because of climate change over a time period. So what I have done here is I've used that exact same model, and then I've used climate scenario, the Canius M2 model, and predicted what these distributions will look like over a couple of different time periods. So this is what it's going to look like in 2025. This is in 2055. And then we have 2085. So it definitely shifts. And to better give you a better idea, I created a surface that looks at the difference from our current prediction, so 2020, and what is the difference between it and 2085. And so here, the really dark blue is areas of the landscape that was more suitable to lodgepole pine, and it's now shifting more towards jack pine genetics. So it's actually a shift from lodgepole to hybrid pine. And then in the dark green, we're having a shift towards lodgepole. So what's happening here is these are areas that right now are suitable for jack pine, but they're predicted in the future to be more suitable for hybrid pine. So what this is actually telling us is there's an increasing range potential for hybrid pine primarily in Alberta and up into the Northwest Territories. And so the idea of using pine as part of a climate change mitigation has been brought up in the literature, and it's really interesting to see how these hybrid pine might do better in future changing climates. But I think it's also really important because what it does is it suggests the importance of considering mountain pine beetle dynamics in hybrid pine. Because if in the future the range of jack pine shifts into hybrid pine, then we need to know and understand mountain pine beetle dynamics in this hybrid pine. And then the final one is also using this. So I've kind of got two main examples and then three examples within this first one. So the final thing with looking at this hybrid landscape is the question is, are all hosts created equal? Well, we all know that mountain pine beetle are less attracted to non-co-evolved hosts, and all hosts are not likely equal for this system. So introgression in this mosaic hybrid zone is going to vary. So what mountain pine beetle is encountering on this landscape will vary quite quickly in space. So a beetle might, be born in, I'm going to say born, that sounds odd, but and we're just going to go with that, be born in lodgepole pine, and within 2 kilometers of that lodgepole pine, as they disperse, they might hit pure jack pine, or they might run into hybrid, or you could have hybrid pine that's more lodgepole-like or more jack pine-like. So we can use the complexity of this system and maybe understand a little bit more about spread dynamics. And so what I've done is I've just zoomed in on a little bit of Alberta where mountain pine beetle is kind of at the eastern edge of its expansion right now. And I've included this predicted hybrid mosaic landscape. So in the dark blue, we have pure lodgepole pine predicted. In the light blue, we have lodgepole-like hybrids. In orange are our 50-50 hybrids. Light green is jack pine-like hybrids. And then in the dark green are pure jack pine. What I'm going to be doing, I was actually hoping to present some of this analyzed, but for the month of January, I was a kindergarten teacher as well as doing my research. So I didn't get this all done in time. But I think what's really interesting is if we look at the pattern of mountain pine beetle attack data, and that's in black, we can see that it's clustering in the more lodgepole and lodgepole pine-like trees. There could be other reasons why. This is a purely qualitative observation, but we can use this hybrid landscape and this hybrid prediction to test some of these hypotheses. So we can develop a better understanding of eastern spread risk of mountain pine beetle using this hybrid mosaic data set that has been developed using genetic ancestry. And so we really want to answer the question, can jack pine support mountain pine beetle epidemics? So maybe it goes into jack pine, but can it spread from jack pine? So that's using a really simple means of operationalizing genetic data. So just using one metric, which is genetic ancestry. So one value, very simple. But as Rhiannon showed a couple weeks ago, It's a lot more complex than that if we want to start talking about resilient loci. And so what we've done so far is we've been able to map really well one genetic metric, but now we have multiple. So for example, if we look at the data Rhiannon provided, we have 26 loci that are different between attacked and unattacked trees. And those 26 markers probably have spatial variation across the landscape. And that spatial variation may not be exactly the same for all of them. And so how do we summarize all of the information across these markers to a simple parameter that we can use to assess range-wide genetic resiliency? So one of the interesting things about both lodgepole and jackpine is, from a population genetics perspective, there really isn't a lot of genetic population structure. So previously, I used about 400 genetic markers and looked at, lodgepole pine on its own, and looked for signatures of genetic populations, and I looked at jackpine, and it was really weak. And so this is kind of the summary of that analysis. So in the lodgepole pine, there's evidence of possibly 2 populations, but I want to note the amount of black within these pie charts. So the colors denote the 2 different populations, and black denotes unassigned individuals. So these are individuals that could not be assigned with confidence to either of these blue and orange clusters, which suggests there's very weak population structure. Again, jackpine, very similar. Most of the structure that I did observe here was because of introgression from jackpine into lodgepole and vice versa. So there isn't a lot of population structure. And so overall, genetic markers don't tend to vary. So we were wondering if we could even find spatial variation that would be informative for management of these forests and understanding spread risk. So what we did for the 26 markers is we looked at them, we looked at just a single parameter. So we counted the number of alleles associated with resilience in each individual, and we plotted that on the landscape. And it wasn't very informative. It was all very similar in color. And so we went back to the drawing board. And so I'm going to present some work that Rhiannon and I have done together that's really new. We put together this map just about a week after she presented. And so of those markers that she's identified, we've kind of selected the top 14 that show strong differences between the attacked and unattacked. So on your right, there are 3 genetic markers. So we have A is for attacked trees and U is for unattacked. The 2 colors denote 2 different alleles, so allele 1 and allele 2. And their frequencies are noted on the y-axis. So the top 2 markers, you'll notice that in the attacked trees, the green allele is at a much higher frequency, and the pink allele is what we would call the minor allele. So it's got a lower frequency, so it's the minor allele. Whereas in the unattacked trees at that first locus, the pink allele becomes the major allele and the green allele becomes the minor allele. So we're looking at a switch between the attacked and unattacked of these alleles from when they become a major allele to a minor allele. So there's strong differences. So I've just given you 2 examples on top of what that looks like. And on the bottom would be a marker we wouldn't include, because in both the attacked and unattacked trees, in that bottom graph, the green allele is the major allele. So we didn't use those ones. So we picked the top discriminating markers. So the ones that really showed a big difference between the attacked and unattacked. And we looked at each individual in our data set, and this includes lodgepole, hybrids, and jackpine. We counted the number of resilient associated alleles in every individual and just plotted the proportion of that. So how many alleles are resilient in this individual, and we plotted it. And then I created a surface. And this is what the surface looks like. So what I'm showing you here is in this, the kind of reddish orange color is where we have a high number of resilient alleles in individuals. And then as we go into that dark blue color, we have a low number of resilient alleles per individual. And so this is an interpolation using a nearest neighbor method because of the kind of spatial inconsistencies. So we don't have a nice grid of samples. So samples are in black, so you know where the data is coming from for the interpolation. And so I'm just going to highlight a couple of really interesting features of this that I think could be interesting for future management of mountain pine beetle and talking about, eastern spread risk. So the first off is we see a pretty good transition in this area from trees with a higher proportion of resilient alleles to trees with a lower proportion. And this corresponds with that hybrid zone. So we're transitioning from lodgepole pine into jack pine. So we're gonna get a pretty big change in this area. The second spot I wanted to point out was here in Saskatchewan, in southern Saskatchewan. I thought it was really interesting that there's a higher proportion of resilient alleles in what I think, Rory in the future can correct me if I'm wrong, but I think these are where the island pines are. And so I think those pines have been a really interesting population in general for forestry management. And then finally, when we get all the way over to Ontario, there also seems to be a little pocket of an area with some higher level of resilient alleles. So the idea of creating a map like this is now we've gone from telling you genes matter to saying, here's how they vary on the landscape. How can we use this? And some of my thoughts is that we can use this to test different hypotheses. So we can say, are these resilience alleles associated with clinal variation? So we can start to look at the relationship between the proportion of resilient alleles and the environment. A tree that's better adapted to drought stress might be more resilient to mountain pine beetle. So those sorts of patterns might come out. The second question is, does resilience partly explain mountain pine beetle spread dynamics? So we have quite a bit of mountain pine beetle attack data from Alberta forestry. Can we use some of that attack data to test this hypothesis? So looking in Alberta, where we have attack data, where we have this prediction, can we look at the spread of the beetle and how it's moved across the province to say whether or not resilience is helping to explain some of these spread dynamics? And so where are we going from here in terms of, this type of research? First of all, I want to increase the resolution for that mosaic hybrid zone. So the zone I've showed you that Ian developed, it was developed using 40 genetic markers across the pine genome, which sounds like a pretty decent amount. But if you think about the pine genome if you think of each base pair in the pine genome as a word in an encyclopedia, there would be equivalent of 464 entire Encyclopedia Britannica's worth of words to the number of base pairs in a pine genome. So if you think about 40 words in 454 entire complete volumes of Encyclopedia Britannica, we haven't really looked at much of the jack pine genome or the lodgepole pine genome with this hybrid ancestry question. So our estimates of ancestry are not likely as accurate as we could make them, which could be really important for understanding mountain pine beetle spread risk. So the next step is using the data that Rhiannon's generated and creating a new hybrid ancestry map from that and comparing it to the current map. So how well have we done with these 40 markers? Does it look like what we find with 10,000 markers. So are we estimating this hybrid zone accurately enough before we start using it in models, right? We can use it right now in models, but if I'm doing this really detailed look at the hybrid landscape and mountain pine beetle spread, I want to make sure it's right. So that's the first step and where we're going from here. The second one is, I think we really need to now take this genomic information and model our spread risk. So can we, hopefully you can't hear my cat sneezing in the background, apologize. Hopefully we can now, take these predictions of where our genetic resilience populations are and say, hey, this does make this does make a difference to mountain pine beetle spread modeling. But also then it can be operationalized. If we can demonstrate that, then if mountain pine beetle enters into Saskatchewan, we know that the stands located centrally in Saskatchewan are more at risk than the stands located in the southern part of the pine range in Saskatchewan. So we can use that to then be more efficient with how we distribute resources. So we can say these stands are more at risk, we need to manage them. These stands are least at risk, they're our second priority. So it's both better understanding mountain pine beetle spread risk, but also operationalizing resources for management.